Hello, my name is Emmanuel Manzabayo from Human Nutrition and Dietetics with registration number 333000351. I'm going to present with a immunological and nutritional aspect of disease, assignment, presentation. I'm going to start with discussion of the following question. The first one is, what are primary function of immune system? The second is, what is the difference between innate and adaptive immunity? The third is, how do T cells recognize and respond to pathogen? The fourth is, what is the role of antigen presenting cells in immune activations? The fifth is, what is the function of major histocompatibility complex MHC? The last one is, how do antibodies contribute to immune defense? The first question was the primary function of immune system. As we know, definition of immune system is defined as a group of complex defense response found in humans and advanced vertebrates that helps to repel disease-causing organisms or pathogens such as bacteria, virus, fungi, and parasites. Immune system also, it is composed of various cells, tissue, and organs that work together to recognize and eliminate foreign substances from the body. Immune system they have two components. There are cells which are T cells, B cells, and macrophages, and molecules, antibodies, and cytokines. The primary function of immune system are recognition, which to identify the foreign substances. Where they work, the immune system uses receptors to distinguish self from non-self. Here is response. In response, there is elimination or neutralizing pathogens, where innate, there are rapid, non-specific, and adaptive response, there are specific. Memory. In memory, they are retaining information from the efficient exposure response. Mechanism. T cells and B cells are generated infection or vaccination. Here there are mechanisms how immune system work, where there is immune response, how immune response are produced. Here we have pathogens which can affect infected cells. Their cells are infected. Innate immune response. Here they provide immediate response, while for adaptive they delay a response. The second question is the difference between innate and adaptive immunity. Adaptive immunity, the response are immediate, while for adaptive is slow. In innate immunity, there is non-specific, while for adaptive is specific. In innate immunity, there is no long-term memory, while in adaptive immunity, there is long-term memory. Both the mechanism, they involve different mechanism. In innate immunity, there are physical barriers, while for adaptive immunity, there is coronal selection. In innate immunity, there is phagocytosis, while for adaptive, there is antibody production. In innate immunity, they use complement system and inflammation, while for adaptive immunity there is immunological response. Here is the comparison between innate and adaptive immunity, where in adaptive immunity there is B cells, antibodies, and T cells, while for innate there are macrophage, neutrophil, dendritic cells, eosinophil, and the basophil. Here both innate and adaptive immunity, they both have similar natural killer cells. The third question is how T cells recognize and respond to pathogen. T cells, they involve in recognition, where T cell receptor binds antigen on antigen presenting cells, major histocompatibility C restricted. They produce response CD8 plus T cells, they destroy infected cells. CD4 T cells, they activate other immune cells. While for regulatory T cells prevent autoimmunity, where they use perforin, coenzyme, and cytokine release. Here there is a mechanism how T cells recognize and respond to pathogen. We have CD4 cells and CD8 cells. Here we have in CD8 we have MHC1, major histocompatibility class 1, and T cell receptor cells. They involve in cytotoxic T cells to destroy the infected one. They involve in two mechanisms in CD8, where when the cytotoxic T cells contract with the MH1 epitotic complex on infected cells, it produces granzyme and perforin. While in CD8, when CD4 plus helper T cells binds MHC class 2 antigen complex on the antigen presenting cells, both the antigen presenting cells and T cells, both they release granzymes. Here I move to question 4. What is the role of antigen presenting cells? Antigen presenting cells such as dendritic cells, macrophages, and B cells play a crucial role in activating T cells. They have a function of capture, process, and present antigens to T cells. Mechanism: first, they ingest pathogen. The ingested pathogen, they are processed antigens. Then those antigens, they are presented via major histocompatibility molecules. This is how antigen presenting cells work. They present antigen to cells. Here is the structure of antigen presenting cells. They produce CD4 mature helper T cells and mature cytotoxic T cell known as CD8. The fifth question is function of major histocompatibility complex. Major histocompatibility complex molecules are critical for presenting antigen to cells, as we saw in the last. The major histocompatibility, they have two classes, which is class 1 and class 2. They present all nucleated cells and display endogenous antigen, for example, viral proteins, and two cytotoxic T cells, CD8 positive. Where in class 2, there is found in antigen presenting cells and present exogenous antigens, for example, bacteria, to T helper T cells. The significance is intra-immune system specificity. For the immune system to be specific, there is antigen presenting cells. Let's move on. That's the question. Sorry. Mechanism how MHC works. There is infected cells. First antigen associated with MHC molecules. Two T cells recognize there's a combination. There is cytotoxic T cells, and another is T cell receptor. The next question is how antibodies contribute to immune defense. Antibodies such as immunoglobulin are glycoprotein produced by B cells. They play several roles such as neutralization, opsonization, complement activation, and agglutination. For the mechanism how they work, antibodies produced by B cells, they produce long-term immunity via memory cells. This is how antibodies work. Here the structure of antibodies produced against toxin antigen, and for example for neutralization, here is agglutination, where there is a grouping of antigenic cells, and precipitation, if a soluble antibody complex is too large to stay in the solution. That is the end of presentation. There is a reference where you can find more information. Thank you.